So hello friends, we are back to the biochemistry classroom. In the previous video, the previous part, we have started the gluconeogenesis, the fundamentals of gluconeogenesis. Okay, so today in this short video I will end the process of gluconeogenesis. So I hope yesterday you remember that I had taught you three enzymes or four enzymes which were bypassing this irreversible reaction so that we could make glucose. Okay, so the main challenge for us was, or the main challenge for the main challenge for the gluconeogenesis is to overcome those irreversible steps so that it could reach the glucose molecule, because the process of gluconeogenesis is the process where you are forming a glucose molecule. You are not metabolizing glucose, but you are doing an anabolic reaction. You are doing an anabolism, in that sense I can say, or making something. So anabolism, you are not doing catabolism. Okay, so glucose is being produced. So we had these four steps: glucose 6-phosphatase, we have fructose 1,6-bisphosphatase, we have pyruvate carboxylase, phosphoenol pyruvate carboxykinase. This all yesterday we have discussed. So we had stopped, I think, if I take a look on this cycle, we had came out from the mitochondria into the cytosol as malate and getting converted into oxaloacetate, and now we are in the cytosol, that is we are in the cytoplasm. We are in the cytoplasm. And to come to the cytosol, you need to make yourself into a malate from oxaloacetate. Oxaloacetate is not able to come out through the inner mitochondrial membrane. So what we do is second thing I told you in the last video is, don't consider this as a malate shuttle, because you are not producing an ADH molecule, rather you are actually utilizing it inside. But once you come out in the cytosol, there from malate to oxaloacetate, you can produce an NADH molecule. Yes. So while coming from this malate to PAP, if I take this part of the reaction, here, I will say to oxaloacetate. So in this process, we are producing what? NADH. We are producing NADH. Correct? Second concept to remember about this thing is that you are in the cytoplasm. So the problem is not anything. You have come into as an oxaloacetate. This oxaloacetate, if you want to convert it into PEP, you need an enzyme, and that is called PEPCK. Very nice answer. So that is called PEPCK. What is PEPCK? Phosphoenol pyruvate carboxykinase, which will convert this oxaloacetate into PAP. Correct? Do we remember in the glycolysis we were converting phosphoenol pyruvate to pyruvate by the enzyme pyruvate kinase, which was an irreversible step of glycolysis. That's why we are bypassing that route. If you can see, we are bypassing that through coming by via the malate and then oxaloacetate, and then I am converting myself again into phosphoenol pyruvate. So you have actually, that irreversible step of glycolysis you have bypassed actually to reach the phosphoenol pyruvate, because you have to go into a reverse direction. Correct? So this 2-carbon compound can be produced from oxaloacetate, which is a 4-carbon compound. So you will be doing a decarboxylation reaction. With the production of GTP. So in this reaction, you will be writing that you are having CO2 and GTP. CO2 and GTP. So you have to remember this point. Okay. So GTP. And you are getting a phosphoenol pyruvate. Rest steps you can understand. Rest step will go as you know. Okay. So next step is that irreversible. Yes, that enzyme I will be using. That is what? Fructose 1,6 bisphosphatase I will be using, because I have to destroy this fructose 1,6 bisphosphate by the ACE, isn't it? By the TASE to form phosphate, isn't it? So I am coming back to this F6P. You should analyze this thing. Okay, so you have to come from this point to this point. In glycolysis, you were going through. In glycolysis, you had to go like this in a blue arrow fashion. But now you have to reach to a glucose molecule. So how you will reach? You have to take a phosphatase enzyme to break this fructose 1,6 bisphosphate into fructose 6 phosphate. So you are going into reverse. In the glycolysis, you were coming from glucose 6 phosphate, fructose 6 phosphate, fructose 1,6 bisphosphate, phosphanol pyruvate. This was the mechanism. This was the way you were metabolizing the glucose. Okay. Okay. Correct. So next enzyme which you have is the glucose 6 phosphatase here, because you have again a phosphate and you have to break this glucose 6 phosphate to glucose, and now you are getting a glucose. Now my question is that what is the location of this glucose 6 phosphatase? What is the location of this glucose 6 phosphatase? Yes, guys. So liver, it's okay. It's okay. What is the location? Cytoplasm. So here I want to make you, because this is what happens when you are reading the books and you know, yeah. So the concept is that I want to add a concept for your good self, is that uh, basically this glucose 6 phosphatase is not kept in the cytoplasm. Remember this concept. It is not kept free in the cytoplasm. This is what conceptual biochemistry is here, because everyone, whenever I ask this question, where is the enzyme, you say cytoplasm. So you have to remember now a fact about this enzyme and a common sense also. See, this glucose 6 phosphatase is not kept, directly in the cytoplasm. Why, sir? You are saying that all reactions are happening in the cytoplasm. Where is it? The cytoplasm, if you keep it in the cytoplasm, what will happen? It will rapidly convert this glucose into metabolites. It will start the process. Okay. So it will convert it into, it will use it, isn't it? Are we able to understand? Because you don't want, you are doing a gluconeogenesis. So do you want it, do you want to keep it in the cytoplasm? No, you will keep it in the endoplasmic reticulum. So in how, no, by which way is it kept in the, you are saying at the same time the cytoplasm, but where? So endoplasmic reticulum. So for that purpose, I want to draw a diagram for you. I want to draw a diagram for you. Okay. So to make this concept of why this is kept, how it is kept, so I will draw a diagram for you. Okay. So let me come to the diagram. So guys, I have made a diagram for you without wasting your time. So you should but make this diagram. Okay. So I have drawn this diagram. I stopped the recording for some time. Okay. So guys, if you can appreciate, the blue is my ER. This is the endoplasmic reticulum membrane. Okay. This is the ER membrane. And inside I have written, this is the endoplasmic reticulum lumen. Okay. So can you appreciate that here it is cytoplasm? Yes, here it is cytoplasm and this is Z. So can you see the glucose 6 phosphatase enzyme which I have made is not present in the cytoplasm. It is towards the cytosolic side. No, it's towards this membrane side, not cytosolic, I should say. I would rather say it is on the membrane, ER membrane. Okay. So what is happening is it is not kept directly in the cytoplasm. It's not directly in the cytoplasm. So that means if my glucose 6 phosphate from this diagram, because you suddenly get lost to somewhere. Okay. So for you people, we have to convert the G6P into glucose. What will I use? Glucose 6 phosphatase. So this glucose 6 phosphatase is present here. This glucose 6 phosphatase is present here, green color enzyme. Okay. So this glucose 6 phosphatase, that means the glucose has to come, has to pass through this. Like, have you seen the factories? What they do in the factory work? What do you see? You see some zones. So at some zones there are people. Okay, people are kept at every zone. So this, for example, if you are manufacturing a mobile, this mobile phone screen will be adjusted by the A person. Buttons will be adjusted by the B person, and then C person will finally give it a, it will put that stamp and everything, the company logo and everything. So this will be the finalization step. Same way, I am saying, once you have this glucose 6 phosphate happening in the cytoplasm, it is per se in the cytoplasm, it is going to, it has to come inside the luminal side of the endoplasmic reticulum. Because sir you are saying that, glucose 6 phosphate transporter is inside, in the lumen, in the membrane of the endoplasmic reticulum, so there should be a transporter. So yes, we have a transporter in the endoplasmic reticulum, that is G6P transporter. It is also called T1 transporter. So G6P1 transporter, where is the this? Yeah, so G6PT1. So this is the transporter through which your glucose 6 phosphate will come. Okay, so it will come like this. So it has to come inside, correct? So here glucose 6 phosphate will be acted upon by glucose 6 phosphatase, and that will break this into inorganic phosphate and glucose. Correct? Has the reaction happened? Yes, you wanted actually a glucose from glucose 6. Guys, is this clear, guys? Guys, is this clear? First, give me a thumbs up or something that you have understood this. Very few people understand this concept, but it is easy. I have drawn a simple diagram for your understanding. Correct? Now, what will happen? Can you appreciate? I have drawn a black arrow in the endoplasmic membrane. So see, I should have drawn another arrow for this phosphate also. Okay. So this phosphate is also going through a transporter. Okay. This phosphate also goes through a transporter. So what happens, this glucose, why, see, glucose has to come in the blood because you are making glucose for giving this glucose to the starving condition of your body, so it should come into the blood circulation. So this glucose has to cross this glucose transporter. This black color is called glucose transporter and is named as T2. So T2, so glucose 6 glucose transporter, glucose transporter, glucose transporter. Okay, so my glucose transporter is present in the this membrane. So this has a transporter. So my glucose, this glucose, this will come through this and is finally is coming out from this. Okay, is coming out from this. Correct. And I hope you remember I had taught you that we have GLUT2. So this is GLUT2. This is GLUT2. Correct. So through this GLUT2, it will come out in the blood circulation. It will come in the circulation. My task is done. Sir, it came in the circulation. And it will cause, what it is going to cause? Hyperglycemia. It will increase the glucose concentration in the blood. So I am having an increased glucose. What about this inorganic phosphate? It is also coming through its own transporter for a knowledge-based question. So this was what was the name of this transporter? It was glucose. This black one was glucose transporter and T2. So this is level T2. And this third one is for the inorganic phosphate coming like this. So this is T3 transporter or inorganic phosphate transporter. Inorganic phosphate transporter. So that's why it has kept. If it is kept in cytoplasm, it will start the metabolism. So did you found what happened? How gluconeogenesis started? So if I locate this region, I saw first it was a mitochondria. It was a mitochondria, cytoplasm, and then ER. So can I make a sequential arrow for you? So sequential arrow was first I had a mitochondria. First I had a mitochondria, and then it went to, yes, cytoplasm. It went to cytoplasm. Very good. And then it went to endoplasmic reticulum. It went to endoplasmic reticulum. Now, can I ask you the glucose transporter? Glucose transporter, can you locate black? What is this orange, by the way? Orange I have made. This is a plasma membrane, guys. This is a plasma membrane. I hope you know the physiology that we have some compartments. We have extracellular compartment, we have intracellular compartment, we have interstitial compartment. All have been taught by me in the physiology, and I have even discussed the clinical things, how you give the patient in the hospital normal saline, why you give normal saline, Ringer's lactate, Hartmann solution. I have discussed this. That is a part of surgery actually, but I have taught you that in the basics, foundation of that. Okay, so plasma membrane is, so this plasma membrane should have a glut that is called GLUT2, so that glucose can come out and cause, help the starving tissues in the body. Uh, a concept arises from this concept. It means that the T2 and T3 transporters, these two, they should get activated in the fed state or fasting state. T2 and T3. I have a question. When should they? Very good. So can I say T2 and T3 are stimulated by fasting state? Yes. So fasting state are actually causing, exactly, exactly. Yes. That is what I had taught in the concept. So that was the concept, guys. So if I tell you location of glucose 6 phosphatase dash, location of glucose 6 phosphatase dash, endoplasmic reticulum membrane. Reaction of glucose 6 phosphatase happening in dash, luminal area of the, this is, that's why I wrote here luminal area. Very good. So reaction happened in the lumen, but the membrane is having the enzyme. So it is like the same way I taught you the factory way. So things are coming, they are getting converted into something, and then they can be produced at somewhere else. Okay. So guys, this is the very basic concepts of gluconeogenesis which I wanted to stamp in your brains. Um, well, another thing to remember is from our previous discussions, if you are new to my class or my video, I had taught you a concept of fasting state and a concept of fed state. I said fed state, I will be having stimulation of glycolysis. I will be having stimulation of PDH. So who stimulates the PDH complex? Insulin. Insulin was stimulating my PDH complex. If I talk about my fed state, when I have eaten everything, I have I have insulin, isn't it? So because I am having hyperglycemia after the fed state, postprandial hyperglycemia, I will be having insulin stimulation of pancreas, beta cell pancreas. They will start excreting insulin by that GLUT transporter, and then that insulin will stimulate my PDH. This will stimulate my PDH. But if I talk about the fasting state, but if I talk about my fasting state, what about the fasting state? My fasting state means I don't have glucose, means I have to start using my proteins, my fats. So that means, should I do beta oxidation of fatty acids or not? Should I do beta oxidation of fatty acids or not? Guys, reply to me. Yes, 100%. I should use fats. So that's why I say in the fatty, in the fasting state, I am having a beta oxidation of fatty acids. I am having beta oxidation of fatty acids. So this beta oxidation of fatty acids will yield me dash acetyl CoA. Acetyl CoA, very good answer. Acetyl CoA. But do you remember, if I connect this dot with this, do you remember in the fed state, by the stimulation of pyruvate dehydrogenase complex, you are able to produce, pyruvate is getting converted finally to acetyl CoA. And that acetyl CoA, I was using it for synthesis of fats. So in that place, I was using fat synthesis. Do you remember in fed state your body stores? So can you understand this relation? When I was doing in the fed state, I was actually producing acetyl CoA for the production of fats, for fat synthesis. But in fasting state, I am not having glucose. That means I should start breaking down my reserve of fats. That is, I should start beta oxidation of fatty acid. That means I should start producing more amount of acetyl CoA, and that acetyl CoA should become an inhibitor of PDH complex now, isn't it? Because if I make more acetyl CoA, it should inhibit PDH complex. My PDH complex should not get, okay. So my pyruvate, if I talk about my pyruvate, it is getting converted to acetyl CoA. The more acetyl CoA you make, the more inhibition of PDH happens. PDH complex happens, which was here, PDH complex. Do you remember PDH complex? Yes, we remember PDH complex. Is this clear, my friends? Is this clear? Is this clear, guys? I think people are, enjoying cold drinks and maybe using WhatsApp and everything, talking to their dear and tear and near ones. Yes. So this is the concept. Okay. So this is the concept. So are you able to understand how things are linked? So you should think first. Don't rot learn it. It's easy. Fed state of common sense, start making fats. Fasting state of common sense, start breaking fats. Making fats, acetyl CoA. Breaking fats, acetyl CoA. Is this clear? So, do you remember that this was the pyruvate which I was using for making the oxaloacetate? Do you remember? Which was the first step of gluconeogenesis. The first step of gluconeogenesis by the enzyme dash. From the pyruvate, I was making OAA. Pyruvate is a 3-carbon compound. OA is a 4-carbon compound. By the enzyme dash, pyruvate carboxylase. Pyruvate carboxylase. That means, this should mean that gluconeogenesis is happening, since gluconeogenesis is happening in the fasting state. So that should mean to me that insulin should start the process of gluconeogenesis, or glucagon should start the process of gluconeogenesis. Every child from a lower kindergarten to a 10th class knows that glucagon is every time available. If you don't have glucose, you have glucose in the blood, postprandial hyperglycemia, you will be using insulin. Glucagon, emergency condition, not having glucose, not having starvation condition, glucagon should rise. Glucagon should start the process of gluconeogenesis, producing the glucose from non-carbohydrate moieties. That insulin should be inhibited. So I come to a conclusion that in the gluconeogenesis, glucagon should be stimulated, and insulin should be inhibited. Is there any doubt in this? No doubt in this. So insulin should be inhibited, because I don't want insulin to work. I want glucagon to work to raise the blood sugar level. And that's why this enzyme will be active in the phosphorylated state. Yes or no? Do you remember that story of phosphorylated state? Yes. So phosphorylated state. Very good. So phosphorylated state. Is this clear, my friends? Is this clear? Is this clear? So I hope you have understood this discussion of gluconeogenesis. Before ending this discussion, I hope you remember these T1, T2, and T3. I will highlight this for you because maybe you get a, amaurosis fugax. Okay, amaurosis fugax. We don't want your amaurosis fugax. I will teach you that in the neurology. So T1, T2, and T3. Okay, so T1, T2, and T3. This is the process you should remember. It's a sequence, so it's. I don't think it is difficult. Okay, correct. So what is the energetics? What do you get finally while you are doing a gluconeogenesis? What is the energetics? This is the last part of this. What is the energetics? So energetics, if you understand, I came to know that my carboxylase was using an ATP. My PG kinase was using an ATP. My PEP-CK was using an ATP. So I had 3 people using the ATP. Do you remember? I have 3 people using the ATP. That is PEP-CK. So moment you come across kinase, you know that we have to use ATP. So PEP kinase is using GTP. Okay. So 6 GTP I am using. Carboxy kinase, carboxykinase. It's not creatine kinase, please. It's carboxykinase. Okay. So carboxykinase using how many ATPs to produce? It's using 1 ATP. Okay. I should not write 6 GTP. I am in some other thought. I'm sorry, guys. I'm sorry. Please correct me. I'm in some other thought. So PEP-CK will use a GTP. I'm sorry, guys. Please correct this. Okay. So PEP-CK GTP. Okay. Carboxykinase ATP and PGK ATP. So how many? 3. But we know that we are multiplying that with 2. Do you remember? We always used to take 2. So from our previous discussion. So if I multiply this with 2, how much I get? So I get 4 ATP plus 2 GTP. So that means I use 6 energies. I'm using 6 energies to produce using 6. So guys, in total you are using 6 energies because we are taking 2 molecules. So that means, sir, are we using 6 energy molecules to make a glucose from 2 pyruvates? Yes. So to make a glucose from 2 pyruvates, see, from 2 pyruvates you are making 1 G. You are investing 6 energies. You are investing 6 energies. How 6? Because you are seeing 2. Correct? So this in total becomes 4 ATP if you multiply this by 2, and this is 2 GTP because you multiply it by 2, it becomes 6 energies. Energies I am saying to make life easy because some of you have missed my classes. So 6 energies I am using. This was from pyruvate. Your question will definitely come because sir, you yesterday taught us we have amino acids, we have lactate. So from how many lactate, how many energies will I need to make, a glucose molecule from a lactate? So I have to make 1 glucose molecule and my substrate is lactate. How many energies will I need to make a glucose? So I will need 2 lactate and put on this 6 ATPs. I will invest 6 ATPs. If I invest 6 ATPs when I am using lactate, so I would need 6 ATPs, 2 lactate molecules to form 1 glucose. Correct. So another concept. What about alanine? I have now amino acid alanine. I want to make glucose. How many ATPs will I use? Again 6 ATPs, but I'm writing TAPs. Wow, I'm writing a genetic code today. So this is 6 ATPs given to this alanine to make glucose. So is this clear, the concept? 6 you remember in your concept. 6. I hope you 6 can be remembered. Okay. So making a glucose molecule from 2 molecules of pyruvate requires 6 high energy molecules, or you can say high energy phosphates to make glucose. So is it an energy investment process? Yes, it will need energy investment process. So making glucose from lactate, 2 lactates, 6 ATP is used. 2 alanines, making a glucose, 6 ATP is used. So I would like to give you a review of this cycle of gluconeogenesis. See, this is having a different story. This story is different. Some people are writing, sir, what about this story? This is about glucose 6-phosphatase story. So you should write it separately. It's not in like the process of gluconeogenesis, but due to the enzyme, we discussed the enzyme, why it is not kept in the cytoplasm. Okay, so where it is kept. So I taught you this, where is it kept. Now I'm telling you, this is the one concept you have to remember. This table is gluconeogenesis. You started with the pyruvate. I'm giving you a review of gluconeogenesis in just 5 minutes. Everything will be done because you have watched the yesterday's video. What is the purpose of gluconeogenesis? I want to make glucose. How to make glucose? I would require 4 key enzymes in this pathway. so that I could bypass, you just look at the diagram, so that I could bypass the irreversible steps of the glycolysis. So the enzymes which I use is pyruvate carboxylase, which is the first enzyme, phosphoenol pyruvate carboxykinase, second enzyme, fructose 1,6 bisphosphatase, third enzyme, glucose 6 phosphatase, fourth enzyme, at the end of my gluconeogenesis. And I hope you remember, we need a regulation. We need a requisite amount of insulin glucagon ratio, so that I can maintain the process of glycolysis and process of gluconeogenesis. So you don't want the same time stimulation of glycolysis as well as gluconeogenesis. Either go for glycolysis or go for gluconeogenesis. Body is not doing two systems at the same time. Just remember this. So they will stimulate these processes according to the conditions, circumstances of the body. Third concept to remember is that I require energy, as you have understood. Gluconeogenesis is requiring energy investment process. It is an energy investment process, and I hope you know fatty acid oxidation is doing that. And then we said we have to start this pathway by pyruvate carboxylase. Pyruvate carboxylase is converting my, yes, fast. Pyruvate carboxylase is converting my pyruvate to oxaloacetate. Oxaloacetate will oxaloacetate, it can't come, it can't uh, it's not permeable to the inner mitochondrial membrane, so needs to convert itself into a malate. Malate will come out in the cytoplasm, where I will be producing an NADH molecule. But when I was converting my pyruvate into a malate, I was using what? I was using an NADPH, isn't it? I was using NADH, not NADPH. Sorry. So I was using NADH. So it is not a shuttle pathway, malate shuttle. It is basically conversion to the malate. Then I was using what? ATP molecules, and I was doing a decarboxylation reaction by PAP-CK. I was using PAP-CK for the decarboxylation reaction. Once I'm in the cytosol, isn't it? So this malate was getting converted into PAP, and now this PAP will be reaching what? Finally to the fructose 1,6 bisphosphate, where I will be using another enzyme, that is fructose 1,6 bisphosphatase, converting my fructose 1,6 bisphosphate into fructose 6 phosphate, getting converted to glucose 6 phosphate, where I need an enzyme which is not in the cytoplasm but in the endoplasmic reticular membrane. That story you remember, how it is coming through this process, that part you have to understand here. So we are having a glucose 6 transporter going into the endoplasmic reticulum lumen, where I have this enzyme in the membrane, glucose 6 phosphatase, converting my glucose 6 phosphate into glucose and inorganic phosphate. Now this glucose has to come into the circulation, so I would require a glucose transporter, that is called a T2 transporter, present again in the air membrane, letting the glucose, facilitating it coming out from the mitochondria into the cytoplasm, and at the same time inorganic phosphate also coming into the cytoplasm, and then glucose T2 type of transporter, which is letting this glucose to come out. This glucose will finally cross the GLUT2 transporter and finally reach into the blood, making hyperglycemia or increased glucose level, which is required in gluconeogenesis. And we discussed that we have some enzymes which are opposite of glycolysis. For uh, hexose kinase, we have glucose 6 phosphatase. For PFK, we can bypass by fructose 1,6 bisphosphatase. And we have for pyruvate kinases to pyruvate carboxylase, where we had started this reaction, and phosphoenol pyruvate carboxykinase is the next enzyme. These are all the irreversible steps of glycolysis. That's why we needed these bypass enzymes. Otherwise, all the other enzymes are the same. And we said that the energetics is that we are using 1 ATP. These enzymes are using 3 high energy molecules, and multiplying it to 2 gives us a 6 high energy investment. And you should remember that what is basically a PEP-CK. If it is needing a GTP, what is a PEP-CK? This PEP-CK is requiring a GTP, isn't it? I have taught you it needed a GTP. So that means it is a lyase. It should be a lyase. It should be a lyase. Okay. And I hope I have taught you enzyme commission number 4. Enzyme commission number 4. So guys, suddenly I got an idea. I wanted to end it here. First, tell me, is it okay? Have you understood this process? Have you understood the gluconeogenesis? Yes, very nice. Othlil, I have taught this. So have you understood this? So if you want, I can make you write two things. Differentiate. Okay. So let us make a separate video for this. Okay. So guys, see the next small video for difference between glycolysis and gluconeogenesis. Okay. So guys, I hope that you have enjoyed this discussion. So no, stay connected with me and please keep sharing. Okay. Share it with your friends so that they can also get benefited. Okay. More videos coming soon on all the subjects of 19 subjects of MBBS. Okay. And clinical videos also coming soon. And also if some PG student, postgraduate resident is listening, the videos for their postgraduate courses will also be coming. Okay. So we will be having some separate faculty for this. Okay. So thank you all. Take care and bye bye.